Hi, my name is Marissa Fuss, and this is my Advanced Concepts presentation 4. Okay, to begin, here's some background information. It is important to remember that both DNA and RNA contain phosphodiester linkages within their structures. This type of bond generates a negative charge, which ultimately helps prevent degrading. The reason for this is because this type of bond helps to keep the nucleophilic species from degrading its structure. Therefore, this type of bond is essential in maintaining the structure in both DNA and RNA. Even though these bonds help to prevent the hydrolytic cleaving within DNA and RNA, it is also important to note that DNA has a stronger resistance to this decomposition in comparison to RNA. The reason for this will be explained on the following slide. Okay, so the mechanism for why DNA is much more stable than RNA. DNA is more stable than RNA for the following reason. It's important to understand that there's structural difference between both molecules. The structure of DNA contains deoxyribose sugar, whereas the structure of RNA contains a ribose sugar. Because of this slight difference, the structure of DNA would be considered to be stronger in comparison to the structure of RNA. More specifically, in the structure of RNA, the 2' hydroxyl group will ultimately cause degradation within its structure because it will react with the nucleophilic species. In return, this interaction will ultimately cause the phosphodiester linkages within the RNA structure to break apart from one another. Therefore, since the structure of DNA does not contain a hydroxyl group, it is a more stable molecule. You can see here there is the hydroxyl group and then no hydroxyl group on the DNA versus this is the RNA. Okay, so the process of DNA replication. In order to complete the process of DNA replication, a wide variety of components are needed. Approximately 20 proteins are needed in order to complete this process. The most important enzymes that are present in this process are referred to as DNA polymerases. There are specific DNA polymerases that serve specific functions throughout the course of this process, which are seen into the figure to the right, and all of them will be described throughout the course of the presentation. Okay, so the process of DNA replication. The process is catalyzed and is essential in attaching deoxyribonucleotides to the growing DNA strand. Components needed for the process include deoxynucleoside, 5' triphosphates, DATP, DGTP, DCTP, and TTP, and magnesium. Then the DNA strand is synthesized using a DNA template. Next, DNA polymerases need a primer in order to begin the process. Once the primer is present, the new DNA can be created and the elongation process occurs in the 5' to 3' direction. After that, there are DNA polymerases that are able to correct the sequence if there are mistakes in the new strand that is generated. So here you can see the reactions that are occurring with using DNA polymerases and then how the elongation process happens and then how the actual reaction occurs and what comes off as a result. Okay, so the complementarity of bases. This is another important concept when talking about the process of replication. So the process of DNA replication needs to be completed with high accuracy. Every base that is added to the new strand of DNA during the process of DNA replication needs to be a Watson-Crick complement of the base that is ultimately on the template strand of DNA. The reason that non-complementary bases are not prominent over complementary bases is because the bonds are energetically weaker in non-complementary bases in comparison to complementary bases. And this you'll just see how the bases are actually added. Then A will always go to T and C would always go to G. Okay, so helicase is one of the enzymes that is essential in the process of DNA replication. There are some units that can be identified in the structure of helicase, which you can see to the figure to the right. The double-stranded DNA move through the center of the helicase structure, which you can see, and ATP and PI are released in the process. The helicase hexamer changes its shape, which ultimately helps move the DNA molecule through the helicase structure. Overall, the helicase structure will separate the two strands of DNA so it can be replicated. Okay, so then topoisomerase is another important piece of the process of DNA replication. These enzymes are responsible for temporarily cleaving DNA in order to generate or degrade supercoils that are present within the structure. Type 1 topoisomerases are responsible for the relaxation of the supercoiled structure of DNA. Type 2 topoisomerases are responsible for generating negative supercoils in the structure of DNA. Type 2 telomerase 2 is a gyration in bacteria. Okay, so then DNA polymerase 1 has both polymerase responsibilities and exonuclease responsibilities. Exonuclease is what is responsible for removing a nucleotide that is incorrect from the sequence. There are numerous ways that this enzyme has the ability to know whether or not there is an incorrect base inserted into the sequence or not. First, the incorrect section of DNA will not be able to be linked to either the new strand or will not be able to be paired with the template strand. Accuracy is increased by approximately 1,000 times when DNA is proofread after it is replicated, which you can see right here. This is the process of actually cleaving the incorrect bases. Okay, so now the overall process of DNA replication. DNA replication is a highly coordinated process. The process of DNA replication begins at the origin of replication. In E. coli, for example, the site is called ORIC locus. At this locus, there are 4 copies of a particular sequence that are preferred binding sites for the origin recognition protein DNAA. Also, when DNA molecules bind to ORIC is what begins the creation of the replication complex. DNAB is then used to help unwind the duplex, which you can see in the figure to the right. Single-stranded binding proteins are then used to ultimately prevent the double helix from reforming, which would ultimately prevent the process of replication from occurring. The pre-priming complex is then formed, which is able to undergo the process of replication. Okay, so now primase. Primase then generates an RNA primer, and then you can see that it being right there. It is also important to know that primase is an RNA polymerase. The primer is removed later on in the process. So you have your DNA template, and then primase comes in to ultimately generate the primer. And then using DNA polymerase, you'll have the new replicated piece of DNA from the template. Okay, so now it is important to know that both parental strands of DNA can be used in the process of replication. The replication fork is where DNA synthesis usually occurs, which is you can see right here. One parental strand needs to be generated in small fragments because DNA synthesis has to occur in the 5' to 3' direction. These small fragments are known as Okazaki fragments. DNA ligase is then used in the process of hydrolysis to join these fragments together. This is known as the lagging strand, which you can see right here. The other parental strand is the leading strand because the generation occurs in one motion without the need to stop for any reason, which you see right here. Okay, so now DNA polymerase III is the holoenzyme that is extremely important in the process of DNA replication. Using this enzyme increases the processivity of the reaction. This means that the enzyme has the ability to increase the rate of the series of reactions without having to let go of the substrate. The substrate is only released when the whole series of reactions is completed. Using this enzyme ultimately increases the efficiency and accuracy of the overall process that occurs. The reason that this enzyme is so effective is because of the B2 subunit, which is seen in the figure to the right. The B2 subunit is essential in the process because it can be used as a sliding DNA clamp. Okay, so then for the leading strand. As mentioned before, the DNA polymerase III can generate the leading and lagging strands of DNA at the same time at the location of the replication fork, which you can see right here. For the leading strand, DNA polymerase III starts generating the new DNA strand when primase generates the primer that is ultimately required. The helicase is then used in order to unwind the DNA as the process undergoes. Single-stranded binding proteins keep the strands unwound, and continuous synthesis is completed as the leading strand is completed by DNA polymerase III. Topoisomerase II is also involved in preventing supercoiling from occur, which you can see the process all right here. Okay, and then moving forward for the lagging strand. The synthesis of the lagging strand is coordinated with the leading strand. The composition of the subunit of the DNA polymerase III haloenzyme is important for the overall DNA replication process. There are 2 copies of the polymerase core enzyme that are present. In these copies, there are DNA polymerase himself, a proof-feeding exonuclease, 2 copies of the dimeric B2 subunit signing clamp, and other proteins that are essential in carrying out the necessary process. The entire structure is important because it's involved with helicase DNA B. You can see all these components right here. Okay, so the lagging strand moves through the polymerase site in a subunit of the polymerase III in a 3' to 5' direction. 1,000 nucleotides are added, and then the DNA polymerase releases the lagging strand template. This is completed when the sliding clamp is released. Then a new loop is formed, where a new sliding clamp is generated, and primase is then used to generate another fragment of DNA. The process continues until the entire new strand is generated. This is referred to as the trombone model because the loop changes in size like the movement of a trombone. DNA polymerase I then fills the gaps between the fragments. DNA ligase is then used in order to connect the fragments with one another. Okay, and then that would be the end of the presentation. Thank you.